In this video, we will discuss about skin cancer and what are the biological changes that can help uh, removing or reducing the skin cancerous cells. So these are the experiment done on the melanoma cells that is received from the patients that have skin cancers. And there are three different kind of melanoma cells used for these experiments. These are M727, M525, M1626, and this SH count is known as short hairpin control and short hairpin calreticulum. So basically, cells are uh, transfected with the lentivirus that contains short hairpin control uh, mRNA and also the short hairpin controlled calreticulum mRNA. So what it does is it will down regulate this SHCRT will down regulate the calreticulum production in the melanoma cells, and this SH count is act as a control. So let us start now what this paper has made a point on in this figure. So first they have done the Western blot to see whether their transduction is working properly or not. For that they have done a Western blot of the cells that are transfected with control SH uh, short hairpin control uh, lentivirus, and the cells that are transducted with short hairpin calreticulum lentivirus containing lentivirus. We can see that calreticulum production is down regulated in SHCRT in all the three cells, and they have also quantified this thing uh, through, and they have also quantified the mRNA production of this calreticulum in those cells, and we can see that in SHCRT cells in all the three cases it is down regulated, and all the and in all the three cases it is significantly down regulated compared to the control. Then next what they did is they did a proliferation assay to check whether this down regulation of calreticulum will affect the cells. Down regulation of calreticulum will impact the cell life or not, and we can see that number of live cells in the control is more compared to the number of live cells in the uh, cells that in which the calreticulum is down regulated. And in this figure it is shown for both two cell system. One is M727, another one is M525. And next they measure the colonies that are grown in those cells and compare them with the control, and we can see that the number of colonies that are grown is very less uh, compared to the control in all the three cell system. Next, in order to confirm that there is a change in the uh, uh, cells uh, cell viability, they did two different kind of assays. First one is the cell cycle assay, and next one is the uh, cell viability or apoptosis assay. In the cell cycle assay, uh, for all the three conditions, we can see that the cells in the subG1 levels uh, is increased compared to the control condition. In the M727 condition, there is very less, around 1.2% of the cells are in subG1 level, whereas when the same cells is transducted with calreticulum SHCRT, the number of apoptosis cells or subG1 levels is increased to around 53%. And in M525, some of the cells are already in apoptotic condition, and this is increased from 16% to 41%. And in case of M16 C26 cells, apoptotic cells are very less in the control condition, that is in 0.71%, whereas in SHCRT this value is increased to 35.8%. This shows that downregulation of calreticulum will promote these cells towards apoptosis, thus to that helps to prevent the metastasis of these melanoma cells, as number of these melanoma cells will be reduced whenever the calreticulum down regulation is there in those cells. And this is the quantification in panel F is the quantification of what is presented in the panel E, and we can see that in case of control, the number of cells in G1 phase is more, whereas in case of when the cells are treated with SHCRT, the number of cells in the sub G1 is more, or we can say in the apoptotic condition. Same is the case with the other two cell lines also. The sub G1 level cells is increased in all the three conditions, as we can see here. And this. So next they did another assay to confirm this apoptotic condition, that is to use the zombie dye and annexin B to stain the apoptotic cells. So this data they have presented here for two cells, that is M727 and M1626, and this region is shown as this region represents the live cells, and we can see in control there are live cells, and live cells are more in M1626. And when the cells are treated with the SHCRT, or whenever the calreticulum is downregulated, we can see that live cells are less and cells can be found in other conditions, and these conditions are the apoptotic condition. This one is the late apoptotic condition, and this one is the early apoptotic condition. So cells are more in the late apoptotic condition for M727, and for HH and for M1626 cell lines, the cells are more in this late apoptotic and also in early apoptotic conditions uh, compared to the M1626. And this is the panel H is the quantification of what is shown in the panel G. We can see that the live cells goes down from the control to the case in which calreticulum is downregulated for both the cases. So this shows that downregulation of calreticulum in the melanoma cells, or the skin cancers affected melanoma cells, will lead to apoptotic, or will trigger apoptotic condition in those cells, thus prevent the metastasis or reduce the cancerous cells. Those cancerous cells have thus might be one of the uh, approach that can be used to kill the cancerous cells or reduce the amount of cancerous cells within the patient. So if you need to understand these things about skin cancer and its relation with calreticulum in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.